Chapter 3, the selection of lymphocytes. Okay, so here, what are we going to need to know for the USMLE? So what exactly is it the USMLE requires me to know about the selection of lymphocytes? All right, well, they want me to know the primary lymphoid organs, what are they? What does their structure look like? And just exactly how they function. They also want me to know the ontogeny of T and B lymphocyte cell surface markers. Ontogeny. There's a word that's often repeated. And I want to take a minute to look up what that means exactly. Ontogeny. And that word means it's a branch of biology that deals with ontogenesis. And ontogenesis is the development of an individual organism or anatomical or behavioral features from the earliest stage to maturity. So basically it's just a development procedure, maturation. Gotcha. So how exactly does the T and B lymphocyte cell, B and T lymphocyte cell surface markers, how do they develop? That's good to know. And finally, they want us to know the structure and function of MHC gene products. Okay then, begin from there now. All right, so as lymphoid progenitors, now remember lymphoid, right? How did that happen? You had the pluripotent stem cells, which were stimulated by interleukin-7, which differentiated the pluripotent cells to form or go differentiate into the lymphoid cell progenitor cells, which will give rise to your T cells, your B cells, and your natural killer cells. And the B cells would further mature into your plasma cells. So now, as these lymphoid progenitors develop in the bone marrow, we have seen that they make random rearrangements of their germline DNA to produce the unique idiotypes of antigen recognition molecules that they will use throughout their lives. Well, that should answer my question earlier. So apparently, they start making, well, the B cells start making their IgM and IgD, and they reorganize their structures and have them already presented up there before they even leave the bone marrow, so that they can bind to different varieties of antigens. Same goes for T cells. They too already have their alpha and beta chain of their T cell receptor ready and set before they leave, apparently. Okay then. The bone marrow, therefore, is considered a primary lymphoid organ. So bone marrow is one of them in humans because it supports and encourages these early developmental changes. B lymphocytes complete their entire formative period in the bone marrow and can be identified in their progress by the immunoglobulin chains they produce. That's explanatory. So we learned that primary lymphoid organ is bone marrow because it supports the developmental changes of lymphocytes, and B lymphocytes finish their entire development in the bone marrow. And you can tell how much they've developed based on what kind of immunoglobulin chains they're producing, basically. Okay, so let's move on. Okay, so here we have a diagram. And here we see there's a bone marrow and then there's a periphery. I suppose that would be blood or tissues, whatever. And you have here a lymphoid stem cell matured due to stimulation of interleukin-7, interleukin-7 stimulating pluripotent cells to differentiate into lymphoid stem cells. And right now they have no markers, but then they decide, we're just talking about B cell differentiation here. So they decide to enter into pro-B cells, progenitor cells, and they have Ig heavy chain gene rearrangement. Immunoglobulin heavy chain gene rearrangement. So first thing, you have the heavy chain undergoing recombination of their DNA, which means that TDT is positive. And once that's taken care of, that chain can enter the cytoplasm and from there go and hang out on the cell membrane. Later it can come back inside when the pre-B cell is starting to make the light chain gene rearrangement. And notice this, that light chain gene rearrangement, you have cytoplasmic mu positive. Okay, so that's interesting. So when first you have that heavy gene rearrangement happening, and once that's done, it enters the cytoplasm. From the nucleus, it enters in the cytoplasm. And now the cytoplasm is positive for the mu heavy chain globulin, yeah, or monomer molecule, mu molecule. But while that's happening at that same time, that's when the light chain gene rearrangement is taking place in the nucleus. So then once the light chain, after that, you have your cell, the cell entering into an immature B cell, and that's when the surface is IgM positive. This is interesting. And then once the light chain is also matured, in a mature cell, the light chain will apparently go ahead out into the surface and bind to the IgM and the IgD. And then you have an activated blast B cell, which is producing some kind of antigen, such as a cytoplasmic Ig immunoglobulin. Oh, and look, on the bottom, they tell me what kind of things that they're expressing. For instance, the lymphoid. So you have different stages, right? In the bone marrow, the B cell becomes, there's a progenitor B cell, pre-B cell, and immature B cell. So the pro-B cell is positive for CD19, 20, 21, and also CD40. It is positive for MHC2, it is positive for TDT, and you have RAG expression also happening here. And while it's in progenitor phase, right now we're going to start, the cell is going to start to recombine its heavy chain. And once it has done that and transcribed that and then translated it, it enters the cytoplasm, and now you have a cytoplasm with the heavy chain, the mu portion, ready to go. So that at that stage, the cell, the B cell, is called the pre-B cell. At the pre-B cell, at the same time, you have the light chain recombination happening, and you still have the cytoplasmic mu molecule there, and you have RAG expression, and you notice that the TDT enzyme is starting to taper off because you don't need TDT for because TDT is not inserting random nucleic acids into the light chain in B cells. It does that in the T cell for alpha and beta, but in B cells it only does that for the heavy chain. But at the same time, you still have the expression of RAG gene, which if it's deficient completely, you have SCID. But if it's partially active, it's called ominous syndrome. Okay, so once the light gene has rearranged, transcribed, translated, it enters the cytoplasm, and from there it goes and is expressed right next to the heavy chain of the immunoglobulin of IgM. And that is called the immature cell, B cell. So notice that there is no IgD in the immature B cell. So no IgD in B cells in the bone marrow. Once you have an immature B cell with both its heavy and light chains together, it has IgM, it is MHC2 positive, it is CD40, CD19, 20, and 21 positive. Then it leaves the bone marrow, enters the circulation, and goes into the periphery, where different areas, probably the lymph nodes and the spleen, other mucosal associated lymphoid tissues, maybe just blood. Anyway, it's immature still. Now, to become mature, it has to start expressing IgD, immunoglobulin. And once it does that, then it will start to produce antigen. Now wait, an antigen has to bind the mature B cell, and that will activate that B cells and be considered an activated blast B cell. And in that situation, once it's activated, that B cell is going to specialize and become a plasma cell that will start producing cytoplasmic immunoglobulins. And some of them will become that, and one of them, or a few of them, will become a memory B cell for future foreign attacks perhaps, so that we have something in reserve to attack anybody else who is of similar biochemical structure. And those would be called the memory B cells. And these memory B cells will have they will not have IgM, they will not have IgD, but they will have IgE, IgA, or IgE. So that is key. A mature cell will have IgM and IgD. A plasma cell or a memory cell would have IgG, IgA, or IgE. I don't know, plasma cell might still have some IgM as well. Don't know yet. I guess we'll read about it. But I think we have a pretty thorough understanding. Also note that immature cells no longer have an expression of RAG gene. And obviously, TDD stopped expressing the moment we started light chain rearrangement. Okay, so moving on. Because these gene segment rearrangements occur randomly and in the absence of stimulation with foreign antigen, it stands to reason that many of the idiotypes of receptors produced could have a binding attraction or affinity for normal body constituents. Right, it does stand to reason. Because, you're growing inside the body. When you're doing all these gene rearrangements in the DNA, in the bone marrow, and they're happening randomly, and there's nothing stimulating the cell to make these rearrangements, they might have a possibility of attracting or of attaching to self cells, for instance. So we have to somehow stop them from surviving, because we do not want our body attacking ourselves. So these cells, if allowed to develop further, could develop into self-reactive lymphocytes that could cause harm to the host. Therefore, one of the key roles of the bone marrow stroma and interdigitating cells is to remove such potentially harmful products. Now, cells whose idiotype has too great an affinity for normal cellular molecules, they are going to be either deleted in the bone marrow or inactivated in the periphery. If they are eliminated inside the bone marrow, that is called clonal deletion. And if they are deleted destroyed in the periphery, this is called clonal energy. So in such a way, only those cells that are selectively unresponsive, or meaning they're not going to respond to any antigens that will bind to our self-antigens, as they call it, they are the only ones that are allowed to leave the bone marrow. Okay, now when they say periphery, do they mean periphery as in circulation, or do they mean periphery as something else? Let me find out. Okay. So in a, well, not yet. So immature lymphocytes that are destined to be the cell lineage leave the bone marrow and proceed directly to the thymus. The second primary lymphoid organ dedicated to the maturation of T cells. So they undergo some kind of fellowship, they undergo a further specialization process. This thymus is a bilobed structure, and it is located above the heart. And the thymus has an outer cortex packed with immature T cells and an inner medulla into which cells pass as they mature. So the outside of the thymus is immature. The deeper you get, the more mature you are. So both the cortex and the medulla are laced with a network of epithelial cells, dendritic cells, and macrophages. I suppose they're ready to attack and kill anybody who dares to attack any cell in the body, which interact physically with the developing thymocytes. Okay. So quickly in a nutshell, the primary lymphoid organs are sites of lymphoid cell development. These are where you have the lymphopoiesis. And we know bone marrow is one place where both the T cells and the B cells start off, but the B cells finish off at the bone marrow. The T cells decide to further specialize and go to the thymus, which is also another primary lymphoid organ. Second lymphoid organs would be things like spleen, lymph nodes, mucosal-associated lymphoid tissues. In a nutshell, remember clonal energy and clonal deletion produce self-tolerance. Anyway, clonal deletion is when it happens in the bone marrow, and clonal energy is when it happens in the periphery. And in a nutshell, T cell precursors leave the bone marrow to undergo selection and maturation in the thymus. Now here we have a picture, the structure of the thymus. Okay, so we see here we have a capsule, this pink capsule with trabeculae penetrating into the substance of the thymus, deep into the thymus. And these trabecula are traversed pretty much with blood vessels, which are probably supplying them with these immature lymphocytes that are destined to become future T cells of the body. And from there, these little blood vessels, the thymocytes, the T cells, or T cells, precursor T cells, they come out and enter, first of all, probably, the cortex. They enter the cortex and they go through a series of trainings, and then from there they enter the medulla. And in the cortex and the medulla, we see that there are, in the medulla, we see that there's a dendritic cell. We also see cortical epithelial cells in the cortex. And we also see macrophages in both the cortex and the medulla. And then there are these medullary epithelial cells as well. So they're probably big macrophages. Okay then. So, as the developing thymocytes begin to express their T cell receptors, remember they just got new T cell receptors at the bone marrow, right? So they start to express it like, hey, look, I'm a new T cell receptors. Now they are subjected to a rigorous two-step selection process. So, because the T cell receptor is designed to bind antigenic peptides presented on the surface of antigen presenting cells in the body, a selection process is necessary to remove those cells that will bind to normal self antigens and thus cause any kind of autoimmunity, as well as those that have no attraction whatsoever for the surfaces of APC. So, two things happen here. All these new recruits from the bone marrow who want to be the new T cell have arrived at the thymus and they have to go through selection. First selection would be, hey, are you going to attach to antigen presenting cells? If they do, good. Second thing that they're going to check for is, hey, are you going to attach any other cells other than antigen presenting cells or any other molecule or any other antigens that are really part of the body? And if they do, they are eliminated. So this is accomplished. How is this done? This is accomplished by exposure of the developing thymocytes to high levels of a unique group of membrane-bound molecules known as major histocompatibility complex, or MHC antigens. So these major histocompatibility antigens, they're a collection of highly polymorphic genes on the short arm of chromosome 6 in the human. So chromosome 6 has a gene, highly polymorphic genes, and these are the MHC genes, and I believe they're unique to each human. There are two classes of cell-bound MHC gene products, classes 1 and 2. Both class 1 and class 2 molecules are expressed at high density on the surface cells of the thymic stroma. So the moment they leave the blood vessel that drops them off at the thymus cortex, they are immediately exposed to cells in the thymic stroma with a whole bunch of MHC molecules, class 1 and 2. And we have to see what they attach to or not attached to. Kind of like, let's see who they decide to hang out with and who they wish not to hang out with. So, class 1 molecules are expressed on all nucleated cells in the body, which means not red blood cells, because they don't have a nucleus. But here they say as well as platelets. They are expressed in a codominant fashion, meaning that each cell expresses 2A and 2B and 2C products, one from each parent. Let's go back to this table 131, class 1 and 2 gene products. So over here on the left-hand side says, in a nutshell, it says MHC class 1. What is that? Basically, it's an alpha chain plus a beta-2 microglobulin. And these are codominantly expressed in all nucleated cells of the body. So we have the class 1 gene products and class 2 gene products. So chromosome 6, its short arm has these MHC genes, and MHC1 is there and MHC2 is there. Now, when these genes are transcribed and then translated, they form these products. Like class 1 gene products would be HLA-A, HLA-B, and HLA-C. whereas MHC class 2 gene products would be HLA-DM, HLA-DP, HLA-DQ, and HLA-DR. Now note that HLA-DM is not a cell surface molecule, but it functions as a molecular chaperone to promote proper peptide loading. To make sure that the peptide is loaded and docked on the cell membrane, perhaps. Okay? So you have class 1 molecules that are expressed on all nucleated cells in the body. So HLA-A, B, and C are found in every single cell in the body that has a nucleus, which means it's not found on red blood cells. I don't know any other cells that don't have a nucleus. And they are expressed in a codominant fashion, meaning that each cell expresses 2 HLA-As, 2 HLA-Bs, and 2 HLA-Cs, one from each parent. Unlike when we were doing the recombination, where if one chromosome ends up producing a functional product, the other chromosome is pretty much shut off. And what was that called exactly? That was called, quickly look that up. If it lets me. I believe it was called allelic exclusion. Allelic exclusion. But here, there's no allelic exclusion. Instead, what you have is a sort of codominance. So both of them are expressed. The molecules A, B, and C consists of an alpha heavy chain with 3 extracellular domains and an intracytoplasmic carboxy terminus. So they have a heavy chain and alpha heavy chain with 3 extracellular domains. 3 of them are outside. It has one that is inside the cell, and that's the carboxy terminus. Seems like that's where most of the carboxy terminuses are. They're usually inside, acting as anchors, whereas the N terminus is usually outside the cell. Anyway, a second light chain, the beta-2 microglobulin, is not encoded within the MHC and functions in transport of the class 1 antigen to the cell surface. So you have to make beta-2 microglobulin, and basically that is going to escort the class 1 gene products HLA, A, B, and C to the surface. Okay. A groove between the first 2 extracellular domains of the alpha chain is designed to accommodate small peptides to be presented to the TCR. A groove between the first 2 extracellular domains of the alpha chain. So there were 3 extracellular domains, right? And so between the first 2, there's a little ditch, and small peptides hang out in there, and that's what it's like, the hand that gives and shows, hey, look at what I have, to the TCR receptor of the T cell. Oh, look, here's a picture. And this is a picture of a class 1 major histocompatibility molecule on the left, and an X-ray crystallographic image on the right of class 1 major histocompatibility peptide binding groove. There it is. Let's see. Okay, so look at that. This is probably the carboxy terminal on the inside of the cell. This is the lipid bilayer of the cell membrane. And this is probably all this is the alpha. Look at this, got two double disulfide bonds, and you have three parts of it, right? Three domains, alpha 1, alpha 2, and alpha 3. But look, it's just one whole chain, just forms three chunks. Now the first two, between the first two, there's a little groove. This is where whatever this cell has been chewing and eating, it likes to hold it in here after eating a little, just to show off and look at what I found, look at what I ate today. It likes to Facebook and Instagram everything. Anyway, it hangs out there, and then it can go and show it to TCR, and the TCR can then be the quarterback who tells them, hey, is this important or not important, pitches the ball wherever it needs to go. Anyway, but this entire molecule can't leave unless it is accompanied by the beta 2 microglobulin, because this is what's going to escort it to the surface. Everything in the cell needs an escort. You can't just not have something. Everyone needs to have some kind of either an escort or some kind of chain or sequence that tells the vesicle that it's in where to shuttle that molecule it carries to. It needs a postal code. And here's the crystallographic. Look at that. That's the microglobulin. This is one long chain, and the 1 and 2, alpha 1 and 2, are very close as a peptide binding group. Wonderful. Absolutely wonderful. Anyway, next, let's talk about class 2 major histocompatibility molecules that are expressed also codominantly, which means that both mama and papa chromosomes transcribe, and their products are transcribed and translated, become into proteins. And on the antigen presenting cells of the body, which are basically macrophages, B lymphocytes, dendritic cells, and hey, here's a new one, Langerhans cells. So these cells will transcribe and translate both alleles on both chromosomes to form proteins such as HLA, DQ, DM, DP, DR. And remember, the DM was just an escort protein. It's not really a surface molecule. It's just an escort. Okay, so the molecules are two chain structures of similar length. Two chain structures. Which molecules are we talking about here? MHC molecules. Okay, so duh. MHC molecules are two chain structures of similar length. And so the class 2 MHC molecules, there's like 4 of them. And each one forms two chains, alpha and beta. And each possess 2 extracellular domains, unlike MHC1, which has 3 extracellular domains, and 1 intracytoplasmic domain, just like MHC1. Okay. A groove that will accommodate peptides to be presented to the TCR is formed at the N-terminal end of both chains. Very good. So you have to wonder, why does MHC1 have 3 domains? Probably the third one is to identify it as like, hey, I'm self. All right, so in a nutshell, class 2 MHC has alpha and beta chains. Class 1 had alpha and beta microglobulin chains, right? Beta 2, right? Here we just have alpha and beta chains. Both of them are expressed codominantly. This one, however, is present only on antigen-presenting cells, whereas MHC1 was present in every cell in the body except those that do not have a nucleus. They are even found in platelets. All right, so here we have a picture of class 2 MHC molecule on the left, and X-ray crystallographic image on the right of class 2 MHC peptide binding groove. Look at that. There are 2 chains, quite equal to each other, an alpha chain and a beta chain. You have an intracytoplasmic. Did I say intracytoplasmic or inter? There's a difference. Intracytoplasmic domain. This is inter, this is intra. Anyway, they have a cytoplasmic and an extracytoplasmic domain. They have an alpha chain that continues on and ends. You have a beta chain that continues on and ends. And in between them, they have a little peptide binding groove, right? It doesn't show it here, but it's somewhere here. And that is where it shakes hands with the TCR and say, pleasure doing business with you, or something, I don't know. Within the thymus, each of these IMIT-C products, loaded with normal self-peptides, is presented to the developing thymocytes. Both of them. Huh? Those that have T cell receptors capable of binding with low affinity will receive a positive selection signal to divide and establish clones that will eventually mature in the medulla. So both MHC1 and MHC2 are found in, let's go back a little bit. So remember all these TCRs, antigen presenting cells are found inside this cortex right here. And these antigen presenting cells have both MHC1 and MHC2. And both of them have self antigens in their peptide binding groups. And we have to see how long each new recruit decides to hold their handshake, for instance. How long do they remain bound? Those, so if their T cell receptors bind not so tightly, very loosely, they will be allowed to carry on. But if they get too chummy and want to just hang out with the APCs, they probably won't last very long. Those that fail to recognize the self major histocompatibility, self-MHC unit at all will not be encouraged to mature. So it's a happy medium. You can't have somebody who's like, hey, I'm not even going to acknowledge you exist, but you don't want somebody who's going to just hang on and just stick right to you either. You just want a nice, polite, hi, nice to meet you, move on, kind of, you know? So if you don't acknowledge or you acknowledge too much, you're out. But if you can just be professional and move on, you can move on then to the next stage. So those that fail to recognize self-MHC at all will not be encouraged to mature. And this would also be a failure of positive selection. So positive selection is, I am selecting to see if you bind a little bit. Those that bind too strongly to self-MHC molecules will be induced to undergo apoptosis, which is a negative selection, because these cells would have the potential to cause autoimmune disease. Although immature thymocytes express two accessory molecules on their surfaces, although immature thymocytes express two accessory molecules on their surfaces, right? Remember, they have T cell receptors and they also have CD3, designed to stabilize the interaction between major histocompatibility receptor, well, protein, and the TCR, called CD4 and CD8. As the affinity of the TCR for class 1 or class 2 MHC is evaluated, the cells are directed to express only CD8 if their TCR binds class 1 molecules, and only CD4 if their TCR binds class 2 molecules. So, quick paraphrase. If you don't recognize the MHC, or you recognize too much the MHC, you fail positive selection. If you acknowledge MHC, just a light nod, and move on, you pass the positive selection. And if you fail the positive selection, that's called negative selection. Okay, that's pretty explanatory. So although immature thymocytes express two accessory molecules on their surfaces designed to stabilize the interaction between MHC and TCR, called CD4 and CD8, so they have, the T cells now apparently, they have T cell receptors, they have CD3 and TCR, which activates CD3, and they also now have CD4 and CD8. And CD4 and CD8 are two other accessory molecules on their T cell surface that is used to stabilize the interaction between MHC and TCR. So it's like, you use one hand to shake hand and you use the other hand on the other person's hand to stabilize that handshake. That's what these CD4, they're the other hand, CD4 and CD8. Okay? So it's like they have three hands. One's the TCR, the main hand that doing the shaking of the hands, and then you got CD4 and CD8 coming out from other sides, probably saying like, yeah, it's secure, but we're not going to hold on too long. As the affinity of the TCR for class 1 or class 2 MHC is evaluated, meaning how strongly you bind and not bind, the cells are directed to express only CD8 if their TCR binds class 1 molecules, and only CD4 if their TCR binds class 2 molecules. Isn't that interesting how it happens? So, so it's like you have these on the first person you meet, right? You come into a room as a thymocyte, you got 4 receptors on you. One's the TCR, which activates the CD3, and you have CD4 and CD8. Now, which hand you decide to use to stabilize your initial handshake with the receiving party there, that's going to decide your basically your future. And if you decide to use the CD4 to stabilize, then you would actually end up becoming a CD4 positive cell. And if you end up with a CD8 stabilizing, then you would end up stabilizing your, you would end up becoming a CD8 cell permanently. So that's interesting. So in a nutshell, you have cells with good receptors will receive a positive selection, and cells with useless receptors will receive no positive selection. Cells with bad receptors receive negative selection, and CD4 stabilizes MHC2 TCR interaction, and CD8 stabilizes MHC1 TCR interaction. Okay then. So, right. Okay, so cells with good receptors, positive, meaning touch and go. Useless receptors receive none, bad get negative. CD4 stabilizes MHC2 TCR interaction. CD4 stabilizes MHC2 TCR. What does that mean? CD4 stabilizes MHC2 TCR, and CD8 stabilizes. So there's another thing. So some cells will apparently have only MHC1, and some cells will only have MHC2. And if you're a T cell with your 4 receptors, and the first person you meet is a cell with MHC1, obviously CD8 is the handshake. It's the hand you're going to use to shake with them, and that is going to be your future. You're going to be a cytotoxic T cells. And if the first cell you meet is an MHC2 cell, a receptor with MHC2, you are going to be destined to become a CD4 cells. Pretty explanatory. Okay then, moving on. Okay, here we are. We have here yes. Oh, okay, so here we have another diagram. T cell selection in the thymus. Oh, this should be a nice review then. So here across on the top, we see CD4 positive and CD8 positive thymocytes, fresh from the bone marrow, followed by thymic stromal cells that are already in. So this is obviously a macrophage, it looks like, right? A macrophage has both MHC1 and MHC2, right? And selected cell population. What happens in which situation? That's what we're trying to figure out. Okay, so high affinity for self-MHC class 1. What is going on here? Okay, so here we have, so here comes a cell, a new thymocyte, comes into the thymus, and they meet with a macrophage. And they decide that the TCR ends up binding to this MHC molecule. Now, if this MHC molecule, it looks like an MHC8 here, decides to bind the TCR will also extend out a CD8 hand to shake, but it's too tight. It's just really strongly binding to that molecule. Immediately it's gonna say, no dude, that's too tight, I'm sorry, you can't proceed, and we're gonna have to kill you. That's pretty severe. and basically they have to undergo apoptosis here. So they're eliminated, sorry. You don't pass the program, you know, you're just dead basically. We don't even let you back out into the system to try out somewhere else, no. Now suppose there's another thymocyte, also CD4, CD8 positive. Its TCR also binds with the MHC1 of the macrophage. Obviously, it's going to extend the CD8 hand to stabilize that. But it's like this kind of sort of just shakes it, like binds. There's a low affinity there, and so it's like, okay, I like your handshake. You can proceed. So that would be a positive selection right there. Okay. Next, so now suppose you have another cell, another thymocyte, right? It comes in, sees the macrophage, and it's like, I'm too high and mighty, and I'm not gonna shake anyone's hand. I can survive on my own, whatever reason they have. But it doesn't bind to MHC1. It doesn't bind to MHC2. It doesn't bind to anything. So we're like, well, if you're not gonna be a team player, I'm sorry, you're out. And so no positive selection, it is also killed. Because we need people who are going to be team players. And next, so now we have another thymocyte. And this thymocyte decides to shake hands using its TCR with its MHC2 molecule. And obviously, what is it going to use to stabilize that? CD4. So once that does that, and you have to see how strong or weak that connection is right there. And if it's a low affinity, not so strong, then it will go ahead and say, yeah, well, we like your handshake. We like that you're doing everything. You acknowledge us, but you're not too much, you know. So you can go ahead, dude, you know, positive selection. And then you have another example here where you have a molecule, thymocyte coming in. It shakes hands with the MHC2, uses a CD4 to bind, but it's so enthusiastic, it just says, yes, nice to meet you, and doesn't let go. Really high affinity. No, not gonna work. So there too also say, I'm sorry, we can't take you, too eager. And negative selection happens, meaning that, you know, you gotta go, you know, apoptosis occur. Okay. So this is a nice review of what we just learned. Now let's read on. This selection process is an extraordinarily rigorous one, no kidding. A total of 95 to 99% of all T cell precursors entering the thymus are destined to die there. Oh my goodness, those are really highly sought after jobs, like residency maybe? High-end residency, I don't know. Only those with TCRs appropriate to protect the host from foreign invaders will be permitted to leave to the periphery. CD4 positive cells that recognize class 2 MHC are destined to become helper T cells, and CD8 plus cells that recognize class 1 MHC are destined to become cytotoxic T cells. Lovely, but very selective. So in a nutshell, CD4 cells that recognize class 2 MHC are Th cells, and CD8 cells that recognize class 1 MHC are cytotoxic T cells. Perfectamo. All right, so here we have another example here. And what we have here is a table. And this table is telling me that there are a whole bunch of markers here. What happens before the thymus? Oh, this is talking about T cell differentiation. What happens in the cortex, and whether or not they proceed into the medulla or circulating T cells. Now, here's the thing. All of this selection process is happening in the cortex, both negative and positive. Selection is not happening in the medulla, it seems. So, markers. So, in pre-thymic area, do you have TDT expression of T cells? Yes, for for alpha and beta. What about in the thymic cortex? Apparently, you still have TDT expression. So I suppose all T cells would continue to have TDT expression always, really. RAG expression is only taking place in the initial part in the thymus only. Now, notice, however, where are we? We were actually looking at the table up north. Here we are. Look at that. RAG expression of this gene is happening in the bone marrow for B cells. I wonder if this is only talking about B cell differentiation, however. In T cells, however, we have RAG expression happening only in the thymic cortex. That's an interesting question. When is RAG enzyme being expressed in the T cells as opposed to B cells? In T cells, it happens only in the thymic cortex, and in B cells, it happens in the bone marrow. We still haven't figured out what exactly this RAG expression does based on our reading so far. Now, we understand that, TCR is expressed. Markers means that, okay, can you detect it? I would think that they already have the TCR receptor pre-thymic, but I think what they're talking about here is it detected in the system? And it seems like TCR, CD4, and CD8 is expressed here. Everything's expressed, even CD2, 2, 3, 4, 8, and TCR are expressed throughout thymus, but RAG is only expressed in the cortex, and TDT only until the thymus cortex, also in pre-thymus. All right, summary. The bone marrow and thymus are primary lymphoid organs, agreed, in which the early development and selection of lymphocytes occurs. Self-tolerance is induced by deletion of self-reactive cells in the bone marrow, clonal deletion or inactivation of self-reactive cells in the periphery, which is called clonal energy. T cell precursors move from the bone marrow to the thymus, where they are selected for self-tolerance by exposure to major histocompatibility complex MHC antigens on stromal cells. Class 1 MHC products are two chain structures. They are alpha chains, beta, 2 microglobulin. The alpha chain is encoded within the MHC, and beta 2 microglobulin is not. That is an escort, separately coded. MHC1 products are expressed on all nucleated cells of the body in a codominant fashion. Class 2 MHC products are two chain structures, of which both alpha and beta chains are encoded within the MHC. Right? So for MHC1, there was only alpha chain, and beta 2 microglobulin was a separate gene product. Whereas in MHC2, both were encoded in that same MHC. Class 2 MHC products are expressed on antigen-presenting cells in a codominant fashion, meaning both chromosomes express that, transcribe and translate that gene into a protein. Thymocytes with antigen receptors that bind self-peptides presented in the groove of MHC1 or 2 molecules will be induced to undergo apoptosis. This is negative selection. So thymocytes would do them well to not bind self-peptides in the groove 1 or 2. Okay. Presented, so they should not bind. So MHC1 and 2 are presenting self antigens. So they should feel it and see, is this myself or not? And they say, no, it's not, and move on. That's the thing here. That's the key to take. Thymocytes with antigen receptors that have no binding affinity whatsoever for classes 1 or 2 MHC are not directed to mature further. Failure of positive selection. That is failure of positive selection. Thymocytes with antigen receptors that can recognize altered self are encouraged to clone themselves and mature. This is called positive selection, and then express CD4 molecules if their affinity for MHC class 2 is for MHC class 2. These will become the helper T cells. And thymocytes with antigen receptors that can recognize altered self are encouraged to clone themselves and mature positive selection and express CD8 molecules if their affinity is for MHC class 1. These will become cytotoxic T cells. So remember that one little part there, that when what's happening is that the MHC molecules are full, 1 and 2 are both full of self-antigens. We want thymocytes who are going to be curious enough to investigate and see, hey, what you got there, and look around and feel around on that self-antigen and see, like, oh, is it ourselves? What is it, first of all? Be curious enough to touch and feel it. But if you find out, you touch and feel it, and then you find that, oh, it's self, and you let go, that means you're a good candidate to be a T cell. But if you're not even curious enough to come and approach the MHCs 1 and 2 to see what's there, you're not a good candidate. Or if you come and see what it is, and then you just say, oh yeah, we should just stick to this, that's also bad. So negative selection would be for those who attach and don't let go. And those thymocytes who don't even come and approach the MHCs, they will have failure of positive selection, basically. All right, questions. An 8-year-old boy is diagnosed with acute lymphoblastic leukemia. Remember, that happens in white young boys. And It's usually TDT expressions, right? And what else is there? Oh yeah, they're neutropenic. They are also, they have low, what's it, thymocyte, thrombocytopenia. They have thrombocytopenia, and they have elevated levels of neutrophils, I believe. Um, no, they have thrombocytopenia, neutropenia, but an elevated levels of lymphocytes. And they're young white boys with this disease. And they are TDT positive. That's what you told me. Yep, that sounds right. All right, so let's see. On this kid, there's flow cytometry that is used to determine the immune phenotype of the malignant cells. Flow cytometry, I don't know what that is. The patient cells are evaluated with monoclonal antibodies. Okay. So you have patient cells, you put monoclonal antibodies for MHC class 2, 19, and 34. That's new. And are found, well, we know that it's a B cell. We know it's an APC. And are found to have high levels of fluorescence with all of these markers. They also process cytoplasmic mu heavy chains. Possess. Okay, so why aren't they being expressed, huh? Why are they still in the cytoplasm? I don't know. Maybe because the light chains are not ready, or they're still developing, or the light chains are still getting made, which means TDT is not being expressed. What is the developmental stages of this cell? Oh, okay, so what are the stages? So first of all, we are looking at B cells, right? They start in the bone marrow. There are progenitor B cells, then there are pre-B cells, then there are immature B cells. Because I see mu heavy chains that have formed, I'm thinking they're still in the process of making light chains. And so I would have to say that they are in pre-B cells, not progenitor, but pre-B cell stage, right before immature. Let's see, what do I have? Oh, hello, there it is. Pre-B cells, pro-B cells, mature, no, that would not be in the bone marrow. Lymphoid progenitor, well, we are fairly developed. Immature B cell, no, yeah. I'd have to go with pre-B cells. Let's look it up. Oh, what question? That was D. D. The correct answer is D. The leukemic cells are pre-B cells. They have rearranged their immunoglobulin genes to encode a mu-heavy chain. MHC class 2 antigens are expressed beginning at the pro-B cell stage, as are CD19 and CD20. CD34 is a marker for early lymphohematopoietic stem and progenitor cells, and it functions as a cells-to-cell adhesion molecule. These cells would also have expressed CD10, the common acute lymphoblastic leukemia antigen, which functions as a metalloendopeptidase. Okay. So let me just go and scroll down to E. There we go. So it starts with, well, lymphoid progenitor cells, right? That's what the first thing is. They would not have completed any of the gene rearrangements necessary to create an immunoglobulin molecule. They would be TDT negative, obviously, MHC class 2 negative, CD19 and CD20 negative, and CD34 positive. That's important to note. So if you're a lymphoid progenitor cells, you are CD3, 34 positive. That means B cells have it, T cells have it, and K cells have it. That just means you're a lymphoid progenitor cell. Okay, then you become a pro-B cell. Pro-B cells are rearranging their immunoglobulin heavy chain gene segments but have not yet completed the process. Therefore, they have no completed chains either cytoplasmically or in their cell surfaces. They would be positive for TDT, they would be positive for MHC2, and since they're a B cell already, they decide to be a B cell, they're going to be CD19 and CD20 positive. And since they are a B cell and an antigen presenting cell, they're MHC2 positive. TDT because we're making heavy chains. Okay, then we go off into pre-B cells who have rearranged their immunoglobulin genes to encode a mu heavy chain. The MHC class 2 antigens are expressed beginning at the pro-B cell stage as are CD19 and 20. 34 we already know means just lymphohematopoietic stem cells and progenitor cells. And it functions as a cell-to-cell adhesion molecule. They would also have present CD10, which is specific apparently to acute lymphoblastic leukemia antigen. I wonder if you could do a test to see for if a cell has CD10, and if they do, it means you definitely are. You have ALL. I don't know. Could be something to. I would think so. I don't know. Immature B cells have accomplished both the heavy and light immunoglobulin chain rearrangements and therefore express IgM molecules on their cell surface. However, I don't think they have IgD. They would be TDT negative, CD19 and 20 positive, MHC class 2 positive, and CD34 negative. That makes sense? Now we have mature B cells, which possess the surface IgM and IgD molecules and are capable of responding to foreign antigens. They are TDT negative because, you don't need it past the heavy chain. MHC2 positive, CD19, 20 positive, CD34 negative. Interesting. This is when they become negative because once they become mature, well, so did immature B cells, didn't they? Immature B cells were negative, CD34 negative. So are mature. I guess that means it's time for you to leave the bone marrow and be this. So it means whether you're not in the bone marrow or not, probably. Because note that they are positive in the pre-B cell stage. Okay. Sounds good. Okay, let's go to the next question. Question number 2. The blood from an 8-year-old boy was analyzed, again, by flow cytometry. These cells were treated with fluorescent labeled antibodies to various cell surface markers before they were evaluated by flow cytometry. Okay. Which of the following markers would identify the B lymphocytes in the sample? So what are all the markers on a B cell, basically, is what it's asking. Let's see the options. CD3 is specific to T cells. CD4, T cells. CD8, T cells. CD56, I think that's macrophages. But CD19, definitely B cells. Do you need to read through it? Fine, we'll read through it. Okay. Correct answer is D. The best markers for identification of B lymphocytes are CD19, 20, 21. 19 and 21 form a co-receptor complex. What is that? During B cell activation. So you activate the B cell and it forms a co-receptor. The role of the CD20 in B cell activation is unclear. Yeah, I noticed that was being omitted a lot. Although it forms a calcium ion channel, CD21 is also a receptor for the C3D component of the complement and the Epstein-Barr virus. New information. So CD19 and 21 form a co-receptor. So when they're activated, they form a co-receptor together. CD20 is a calcium channel, basic ion channel, and it is CD21 also binds to the C3D component of complement, and it also binds to Epstein-Barr virus. CD21 is important. Not only does it bind to EBV, it binds to the C3D component of the complement, and it needs to form a co-receptor complex during B-cell activation. CD3 is the signal transduction complex of T cells. It is found in all T cells in association with the T cell antigen receptor. CD4 is found in all helper T cell molecules. CD8 is found in all cytotoxic T cell lymphocytes, and 56 is a marker for human natural killer cells. I was wrong. CD56 is for human natural killer cells. Natural killer cells will also have CD34. Okay, next question. An 18-year-old member of a college soccer team is seen by a physician because of chest tightness and dyspnea on exertion. Sounds like asthma. A 15 centimeter mediastinal mass is detected radiographically. Yikes. 80% of the white blood cells in the peripheral blood are small. Abnormal lymphocytes, they are lymphocytes, but they're really small. I'm thinking we're talking about natural killer cell, maybe. With lobulated nuclei and scant cytoplasm. Lobulated nuclei, scant cytoplasm. Well, now, that is interesting. I know. And you say it's so I have to figure out some kind of a white blood cell that is small. It's a lymphocyte of some kind. It's not. So it can either be beta. It can be either a B cell. It could be a T cell, or it could be an NK cell. And the thing is, they all look the same. And they all have a very big nucleus. Scant inside, well, they could be either. I don't know. Immunophenotyping of the abnormal cells shows them to be CD4 and CD8 positive. Well, then, we know what that is. It's a thymocyte. But it's in the blood. Where would such cells normally be found in the body? Well, I would say they would be found probably leaving the bone marrow and entering the thymus's cortex. So bone marrow is in peripheral blood. Well, normally I would say thymic cortex. That would be my answer. But I'm sure they're just leaving when they become. So I'd go with C, thymic cortex. Let's see how right I am. This might be wrong just because I see. And I said thymic cortex, right? Which was C. Indeed it was. And C is, the correct answer is C. This patient has a T cell lymphoblastic lymphoma. Okay. As opposed to ALL. This is TLL. In his case, the malignant cell is double positive. It possesses both CD4 and CD8. That's not cool. It's like overworking somebody. In a normal individuals, these cells would be found as an early developmental stage in the cortex of the thymus. Once cells have rearranged their receptor genes and been subjected to positive and negative selection, the cells leaving the thymus will express one co-receptor or the other, but never both. Okay, bone marrow would contain T lymphocyte precursors that are double negative. They will lack, oh, I see. So bone marrows at that time, they don't have CD4 or CD8. When do they get those? Peripheral blood would have mature T cells that have differentiated into either helper CD4 positive or cytotoxic CD8 positive cells. There should be no double positive T cells in the peripheral blood. Thymic medulla is the location of maturing T cells ready to circulate into the bloodstream. And peripheral lymphoid organs, it would have only single positive cells. Uh, Maturing T cells ready to circulate into the bloodstream and peripheral lymphoid organs. Only single positive, just like the blood. Splenic periarteriolar lymphoid sheets are the T cell dependent areas of the spleen. They would have fully committed helper CD4 plus or cytotoxic CD8 cells. Okay, splenic periarteriolar lymphoid sheet. This is some place new we've learned. They are T cell only. Around the arterioles, there are these lymphoid sheets in the spleen. That is the place where T cells hang out, ready to be commanded into action, and they're fully committed. Got it. All right. A 12-year-old child is diagnosed with a T cell lymphoma. The phenotype of the malignant cell, T cell lymphoma, the phenotype of the malignant cell matches that of normal progenitor cells that leave the bone marrow to enter the thymus. What cell surface markers would you expect to find on the malignant cells? Okay, well, there will be CD4 negative, CD8 negative, TCR positive. And the only option I see here for that is B. What did I say B? Oh boy, stop now. Fourth is B. Where is 4? What? Negative, negative, negative. Oh man. All right, well, I'm gonna have to stop. That was it for this one.